So far, we've said that DNA is in the nucleus, that we use mRNA to copy it. mRNA is allowed to leave the nucleus, and now we're at the point where mRNA is going to be read and used to make protein. I can't say this enough. If I keep repeating this, it means it's really, really important. The point of all of this is to make protein. Transcription, what we talked about in the last LO, is going from DNA to mRNA. Now we're going to translate it, go from mRNA into protein. Together, transcription and translation make up what is called the central dogma. Now let's look at what happens for translation. Down here we have our mRNA, and we're going to assemble a ribosome on it. Remember when we learned about organelles of cells? Ribosomes make proteins. So the mRNA copied the DNA. It went out of the nucleus into the cytoplasm where a ribosome is now going to chomp down on it. We're going to be using something called tRNA, and tRNA is what is actually doing the translating into amino acid talk for us. On one end of the tRNA we have what's called an anticodon. You see here is UAC. And it matches a codon, which is simply 3 letters grouped together in mRNA. So the mRNA codon here is AUG. A pairs with U, U pairs with A, G pairs with C, codon, anticodon. tRNA is still RNA, and that's why we're using U. So on one end of tRNA we have an anticodon, and the other end carries an amino acid with it. I'm going to continue the animation here. The ribosome chomps down so it assembles, and then we're going to start making a protein. OG is always going to be our start code. So the ribosome is going to find where OG is and bring in the first amino acid, and whenever it's OG, the first amino acid coded with it is always MET for methionine. Okay, I'm going to continue the animation. The next tRNA comes in. If you look, our mRNA codon is AAG, and it brings in the anticodon of UUC because A pairs with U, A pairs with U, G pairs with C. Whenever you have a tRNA that has UUC on it, the other end is always going to be this amino acid, lyse or lysine. Okay, let's continue. As we move down the mRNA, the amino acids are going to get hooked up together. Remember, beads on a necklace. We're hooking up beads and we're making a necklace. The ribosome moves down, opening up a spot here, and then we're going to kick out that tRNA because its job is done. It's going to go get another methionine tacked on it. Okay, here we have UUU. That's the codon on mRNA. So what's the anticodon going to look like? U pairs with A. So the next tRNA coming in should be AAA. It is. And whatever is attached to AAA in terms of amino acids comes along with it. So AAA will always bring in Phe or phenylalanine. The ribosome is going to move down as the amino acids are linked up. And simply amino acid chain is going to make a protein eventually. So we keep going and adding more and more and more until we get a protein chain. So here we're going to move down another one. And we're going to keep going. Next we have an RF. Now usually these chains get really long, but for the sake of this animation they're keeping it short. RF just means release factor. That means that this process is done. The ribosome and everything's going to disassemble, and then our protein can go off and be modified and go do its job. Let's actually go through the steps of translation for the sake of your notes, and then just like we did in LO3, you're going to watch a video by the DNA Learning Center about transcription and translation in real time. So, the site of translation is the cytoplasm. We started in the nucleus, we had a gene that we copied with mRNA. mRNA got sent out, and now we're working out here with ribosomes and tRNA in the cytoplasm. The major players will be mRNA, which stands for messenger RNA. We've already met mRNA. rRNA is what makes up the ribosome, that big thing that chomps down on the mRNA. tRNA is transfer RNA. It has the anticodons and the amino acids on the other side. And our final player is the amino acid chain, or the protein, that we make at the end. What actually happens? So we already did transcription, now this is where we pick up translation. mRNA gets shuttled out to the cytoplasm. The rRNA, the ribosome, chomps down on it and finds where OG is, because OG always means start. That's where we begin. As we continue down the chain, the ribosome has places, you saw this, where the tRNA comes in and fits and matches up with the codons on mRNA. One end of the tRNA has an anticodon, which is complementary to the mRNA codon. It's kind of like a puzzle piece. So on our mRNA, the puzzle piece is UUA, and we have to find what matches with UUA. The tRNA that matches is A, because U pairs with A, U. So this one would come in and bring its amino acid with it. So one end has the anticodon, the other side carries the amino acid. Ribosome continues to move down that mRNA chain. It's going to chomp its way down to the end, reading the codons. The tRNAs keep coming in and dropping off their amino acids, and then they float away to get reset and reused. The amino acids, as they get dropped off, are hooked together by a linkage called a peptide bond. And then once the ribosome reaches stop and that release factor comes in, everything falls apart, the protein goes where it has to go, it might get modified along the way, and then it can do its job. We changed languages now. That's why this is called translation. We started in nucleic acids. DNA was an ATGC. RNA was an AUGC. It was a different dialect, just like if I started talking in a British accent again. Different dialect, but still nucleic acid language. In this case, we completely changed the language. Now we're talking in protein language, which is amino acids, and all of these guys here are amino acids. So this would be like if I started talking to you in Spanish, which I won't because I haven't taken Spanish in a long time and it would probably be awful. But we started in nucleic acid language, and now we've translated the message into the talk of proteins. As I mentioned before, just like we did a transcription, now I'm going to show you that translation video clip from the DNA Learning Center, which shows you this process in real time. If you have any questions, let me know. Look in your note packet so you know what's coming up next.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.